Hello everyone, I am Dr. Vinod Tiwari, currently working as Associate Professor of Pharmacology at Department of Pharmaceutical Engineering and Technology at Indian Institute of Technology, Banaras Hindu University, Varanasi. I welcome you all to this NPTEL course on pharmacological and toxicological screening methods. In an era where drug discovery and development are advancing at a very fast pace, it is very important to learn all the advanced techniques in the field of pharmacology and toxicological screening areas. Whether your path leads to academia, industry, regulatory agencies or clinical research, a solid understanding of how drugs are assessed for their safety and efficacy. So this is very important in shaping the future of drug discovery and healthcare. Over the next 12 weeks, I am going to take you through different screening assays, starting with the ethical principles and guidelines which are associated with the animal handling, caring for them and maintaining them while we use them for our research. Followed after this, we are going to talk about the different in vitro techniques which serve as a very important alternatives to the animal research. Then we'll be also talking about recent computational in silico models which are quite helpful in increasing the pace of drug discovery, and then we'll be also talking about some stem cell and other cell based assays, followed by other technologies including macrofluidic. In the pharmacological screening assays, we are going to learn about the pathophysiology of different diseases such as Alzheimer's, Parkinson's, depression, pain, diabetes and cancer. Followed after this, we are going to learn about the different in vitro and the in vivo models which are widely used for the screening of therapeutics against these deadly disease. Through this, you will gain a critical insight into the new potential therapies, how they are identified, and this knowledge will equip you with the essential. Additionally, we will be talking about the different toxicological techniques including the reported toxicology, carcinogenicity, mutagenicity, along with acute, subacute and chronic toxicity studies. There are molecules which are used in the treatment of different disease, but we do not know about their safety aspects. You might have heard about the story of thalidomide, the drug which was introduced into Europe for the treatment of morning sickness in the pregnant woman, but that has turned into one of the potential disaster in the field of drug discovery, where thousands of kids were born without limbs. So to avoid this kind of situation in near future, it is very important to pay consideration to the different toxicological screening assays well in advance. So we are also going to talk about the humanized mouse models along with other transgenic techniques which are quite helpful in bridging the gap between the basic and the translational science, so that new drugs can come into the market for the treatment of different disease conditions. Whether you are an undergraduate student, postgraduate student, research scholar or an industry professional, this course is going to help you a lot in terms of building a basic understanding about the different pharmacological and toxicological screening techniques, how the different drugs are discovered and how their safety is ascertained before they come to the market. So let's start this wonderful journey together, where we will be exploring step by step how safe and effective medicines are developed through the scientific rigor and innovation. Thank you.